And now we are in Chapter 3, the second part, Part B, which is going to go into the active processes that are going to require energy, and in some cases, a plasma membrane receptor. Okay, so for the active processes, we're going to be talking about active transport and vesicular transport. And in some cases, things are too large or things are not in the right concentration so that you can move a molecule or you can move membrane, but it is going to require an input of energy in order to do that. Okay, so here we have active transport. What it needs are two things. It needs a carrier protein. So this is going to be a receptor, a protein on the plasma membrane, very specific for a very specific molecule. Here's the next sentence. Moves solutes against a concentration, low to high. What I would emphasize is the low to high. I definitely want you to get the concept of concentration gradients, but this is about molecules that are low in one compartment and high in the other. And if you're going to move something from low to high, now they're going to use the action potential as an example of this. I am going to also mention cells that want to store molecules. Because if you want to store something, I'm going to pick on liver cells, hepatocytes, and they have to store glucose. Because if you are, let's say, running a marathon, you're going to run 26 miles, you're not going to stop for dinner. And what you need to do is you need to rely on the storage of glucose, excess glucose, in the liver and skeletal muscle, and you can polymerize glucose into what are called glycogen chains, which are merely like glucose molecules, beads on a necklace. And so then as you're running and your blood sugar gets low, then the body can call on the stores of glucose in the glycogen storage in the liver and in the skeletal muscle. So the person can actually chip away at their own stored glucose during that 26-mile run. To do that, you're going to need to store so that the amount of glucose in the glycogen chains in the liver are very high. And then what do you do before a race? Before the marathon, you eat spaghetti, which is very starchy, which has a lot of glucose. So very easy to digest. So a runner eats their spaghetti before the night before, and they go to bed. And then the body breaks down all of that starch into glucose. And what the runner is hoping is that the excess glucose, because they're sleeping, are going to go into the liver and going to go into the skeletal muscle so that when they start to run, they can actually eat their spaghetti. They can use the energy. And that means that you're moving molecules from a low concentration in the blood, relatively speaking, to high concentrations in the liver and in the skeletal muscle. To do that, you can, but you need receptors for that glucose that is going to take a low concentration of glucose in the blood and transport it into the liver, where you might have trillions and trillions of glucose in chains and in glycogen. So active transport is absolutely essential for those organs that store and are moving things from low to high. That's not the way that it normally works. So this active transport is not going to be for every cell in the body. It's going to be for some cells in the body that want to take molecules in action potential. You're taking, sodium and potassium where they are low in one compartment, and you're going to transport them where they're high in another compartment in order to get that electric signal going. And in all cases, you need a receptor and you need an input of energy in the form of ATP in order to do low to high. Okay, so they're going to use a primary active transport as the sodium potassium pump is part of the action potential. Um, I'll go through these slides. I'm not going to test you. It's not going to be try again. The sodium potassium pump is not going to be on the modal quiz. I prefer talking about storing energy in the liver and the skeletal muscle. But again, that's not going to be on the modal quiz either. But the concept of active transport, moving things from low to high, needing a receptor, needing ATP will be. So here we go. This is a lot about the sodium potassium pump. The reason why I don't like it as an example is that you really don't talk about nervous system until later. And right about now, out of context, it gets just a little bit confusing. So we'll meet this again when we talk about the nervous system. Right now, this is their example in the book of active transport. Okay, co-transport, symport, antiport, not going to be on the modal quiz. But vesicular transport, love that word. So here, what you're talking about is moving whole sheets of plasma membrane. And there are a number of processes that some cells in the body need to do this. Again, not every cell in the body needs to do this. What you need if you're going to have whole sheets of membrane move, and this happens a lot with most white blood cells, which are also called leukocytes, neutrophils, and sometimes they're called phagocytes. And when you do that, you need a lot of ATP energy. And that's the reason why it's called active. You're going to have to import energy in a very active fashion in order for that to happen. Now, you can have vesicular transport do exocytosis or endocytosis. And I am going to, or I want you to know for the Moodle quiz, the steps of phagocytosis. I'm not going to test you on pinocytosis or receptor-mediated endocytosis. Phagocytosis, because it is so important, it's a 24/7 mechanism wherein you're circulating white blood cells that we call phagocytic, and they're the majority, are constantly protecting you against any sort of invasion of a pathogen in the bloodstream. And it saves your life on a daily basis. But phagocytosis comes from the Greek and Latin, which means to eat. And what they're trying to eat are pathogens. And thank God, pathogens, mainly bacterial, are very small. So if you have a phagocytic white blood cell, they're engulfing bacteria that are 10 to 100 times smaller. But on the other hand, if they do not eat and remove them from the blood, you'd be dead within 24 hours. You'd be in septic shock, which can be fatal. So, yay for vesicular transport and phagocytic white blood cells. So let's talk a little bit about this. I want you to think about an amoeba. Amoeba is a tiny little protist that you find in ponds that can constantly change their shape. And phagocytic white blood cells, like neutrophils and macrophages, those are white blood cells in circulation that constantly monitor the presence of pathogenic bacteria. So they are going to, by an amoeba, they're going to change, or like an amoeba, they're going to change their shape. And that's something that's fairly uncommon in the body. The body cells tend to have one shape, like columnar epithelial, or columnar epithelial for the rest of their life. But amoeboid-like phagocytic neutrophils, macrophages, these types of white blood cells can change their shape, which allows them to do phagocytosis. But if you're going to change your shape, then it means whole sheets of plasma membrane are changing their shape, and that is only going to happen if you have ATP energy input. Okay, so this is phagocytosis. This little orangey red ball is a pathogen, and here the cell is engulfing it. Now, do you see the sheets of membrane, cell membrane, plasma membrane, that has to change shape in order to engulf that bacteria? And that is the ATP energy needed, and you have to input that energy first before the cell can actually change its shape and engulf the pathogen in the process called phagocytosis. So here's the steps. This could, hint, hint, be on the Moodle quiz. So there are receptors on the membrane that can specifically attach the pathogen. Engulfment is the act of moving the sheets of plasma membrane to engulf. It's almost like trying to create a vacuole, and the whole sheets of membrane with the input of ATP will allow it to approach that red ball, that pathogenic bacteria, and start to internalize it. Now, once you've internalized it, and right here you have a little vacuole with the pathogen in it, you want to kill it. And that's when the killing is when the lysosome deposits all of its poisons in that vacuole and kills the little sucker. And then the last part is exocytosis. You break it down after you destroy it, recycle any parts you can, but any parts of the pathogen that you cannot recycle is going to then travel to the edge of the cell and be expelled. And that is exocytosis. So you have the attachment of the pathogen, then you have the engulfment of the pathogen, then you kill the pathogen, and any undigested bits get spit out of the phagocyte, and you live happily ever after. So that is it. And we are going to go to part C in a bit. And that is the end of Chapter 3, Cell, Part B.